Right, morning everyone. So I hope that your week's been okay so far and that you had a lovely holiday. Now, today we are going to just be reviewing the vaccinations section that we've done and add a little bit more onto it because we weren't quite finished with it, and look at particularly about how we can apply some of that knowledge. So we're going to focus on what vaccination is again. We're going to look at how vaccines work, how they make you immune to a particular disease, and then about the graph. We're going to review that again, looking at how antibody production changes from the first to the second infection, and then we're also going to have a look at how we can prevent the spread of infection, so this idea of herd immunity. So reminder for you, what we were looking at was that when you first get infected with a particular pathogen, the antibodies on its surface cause your white blood cells to produce antibodies, and those antibodies will work to destroy that particular pathogen. Now, what also gets produced are these special type of white blood cells, which we call memory white blood cells or memory lymphocytes. So these are white blood cells that remember how to produce the correct antibody in order for you to be able to destroy the specific pathogen. So they can remain in your body for years and years and years and even decades. And if that pathogen comes into your body again, then what's going to happen is those memory white blood cells will rapidly produce the correct antibodies and they will help to destroy the specific pathogen. That means that the person doesn't get ill, they don't get sick with the disease second time, and so we can describe that person as being ill. Now, that second time around, remember, we call that the secondary response, whereas the first exposure we call the primary response. Now, we've got a graph here. We did this in class, so I just want you to have a think about this again. What is this graph showing? So describe it, what's happening within these two sections, and then explain it. So, what are the differences and what does that mean? How does that link to what I've just said? So pause the video, have a go at doing that now. You can write that down or you can just do it in your heads. Okay, so what you should be identifying is the fact that we have a much slower increase in this first, in the primary response compared to the secondary response. Secondary response, the increase occurs much sooner, and then the gradient of the line, the steepness of the line, is much steeper in the secondary response. So we have a much faster response, a much slower response here. So a smaller and slower response, a bigger and faster response. So the memory cells mean that we're getting the antibodies produced much more quickly, and we're getting much more of the antibodies being produced because the peak is much higher. And this means on that secondary exposure that you don't get ill. So this is covered within the gap fill that we did within our booklet, but this is it again. So running through the points, when you first encounter that pathogen, it takes several days to produce the correct antibody and enough of the antibody to destroy the pathogen. And that's because the white blood cells need to produce the correct antibody to lead to the destruction of that specific pathogen. Now, our key point is that we end up with the memory lymphocytes remaining in the blood, and so on secondary exposure, when you get the same pathogen infecting your body again, then the concentration of antibodies is going to increase much more rapidly, and you're going to get much more antibodies produced as well. And so that means that you're not going to feel ill, or you're only going to have very mild symptoms, and so we consider the fact that you are now immune to that specific disease. Now that's natural immunity. That's what your body naturally does when you are infected with a disease. So you get infected with chickenpox, you don't get infected with chickenpox a second time. But we can make use of that. And we make use of that using artificial immunity, where we artificially stimulate your immune system in order to make you immune to a disease. And the reason that we do that is because some diseases are so dangerous that we don't want to risk anyone catching them in order to gain immunity. We want to give them prior immunity before they're even exposed to the disease. And so the classic example of this is, of course, smallpox. And we talked about this a little bit in lesson. So smallpox was a very disfiguring disease, very dangerous as well. And so it's been the first disease that we developed vaccinations against. So vaccinations means that you'll become immune. You haven't actually had the disease, and so that if you end up being exposed to the disease, that you won't suffer the effects of it. And so, as we've said, smallpox is a really successful example of vaccination. So vaccinations are all about stimulating your immunity artificially so that we are able to be immune to a disease that's potentially really dangerous for us. So the question is about how these work. So a vaccine, one of the things that a vaccine can contain is small quantities of a dead or inactive pathogen. Now, the bonus of a dead or inactive pathogen is the fact that it has these antigens on its surface, which are the same as the real thing. And so what that's going to do is it's going to stimulate the white blood cells in order to produce antibodies against those antigens. And so those antibodies will help to destroy the pathogen, and so, or the vaccine in this case. And then what you're going to end up with is those memory cells left behind, which remember how to produce those antibodies in order to destroy that pathogen the second time around, and so that we'll get that secondary response. Now, because this pathogen is dead, it doesn't cause the disease in a person. You might end up with some mild symptoms as side effects from a vaccination, but you don't get the actual disease. So what happens is when you get exposed to the pathogen, the real pathogen, the active pathogen in real life, so you're wandering around, you come across someone that's got this and you catch it from them, that pathogen will enter your body. Now, you're going to have these memory white blood cells which have remained in your body from that vaccination that you've received. So in comes the real pathogen. We can see it's got the same antigens on its surface, and so your body is going to respond to this. And so what's going to happen is that those memory white blood cells remember how to produce the correct antibodies, and so it produces those correct antibodies. It produces loads of them. It does them really, really quickly. And so what's going to happen is that those antibodies are going to help to destroy the pathogen, and that real pathogen is going to be destroyed really quickly. And so you're not going to suffer from the disease. At most, you will have mild symptoms from it. So the whole point about this is it is stopping people getting infected with a disease in the future. And what that means is that they are protected from being infected by a particular pathogen. Because their immune system has these memory white blood cells, they're able to produce the antibodies, the correct antibodies, in order to be able to destroy the pathogen. And so that means that you don't get ill. As a result, that means that we can describe that person as being immune. They cannot catch that specific disease. Now, where this becomes really important is not just thinking about individuals, it's thinking about the community as a whole. And so there's this idea of herd immunity, and you'll have heard this talked about a huge amount when we're talking about coronavirus. Now, it wasn't really talked about in the same way there, because they were looking at people catching the disease in order to develop immunity, whereas we're thinking about immunity from the point of view of a vaccination program and what it can do. Now, what we've got to remember is that within our community, there will be people that will be unable to be vaccinated because their immune systems don't work very well. They may have had cancer, and so had cancer treatments. They may have had transplants or something like that. And so there are some people that will not be able to be vaccinated. And so we can protect those people by making sure that a sufficient number of the population is actually vaccinated. And we're talking about a large proportion, looking at something like 85, 90, 95% of the population being vaccinated. So if we have a large proportion of the population that is vaccinated, they will be immune to the disease, they'll have been immunized. And so what that will mean is that the pathogen will not be able to spread through the population, because it will not be able to find people that are susceptible to it, that haven't had the vaccination, and so it won't be able to reproduce itself and it won't be able to pass on to other people. And so what that can possibly result in is the disease actually disappearing, as in the case of smallpox. For the most part, this doesn't happen for quite complicated reasons, but we can protect large numbers of the population. So the real danger about this is when not enough people get the vaccine, and so we don't get up to the herd immunity levels. And so what that means is that portions of the population aren't protected and sufficient numbers are able to catch the disease, and so the disease is able to spread again. Now there are lots of reasons. There's the whole anti-vaxxer kind of movement, which is based on worried about the kind of side effects of vaccines. I've got an exam question for you to look at, which will show you that a lot of this stuff is unfounded concerns. It's not based on the science that we see. And we have to be really careful, and we have to go into these programs with our kind of eyes open. Remember, the key point about vaccination programs are that we are looking to protect the population as a whole, and so we're looking to vaccinate large numbers of people, the large proportion of the population, and what that does is it means that it protects the small number in our populations that are able to be vaccinated, these vulnerable groups. So I want you to pause the PowerPoint in a second. I want you to answer these simple questions, and then we're going to go through the answers. So, what is in a vaccine? Small quantities of dead or inactive pathogens. What does a vaccine cause your white blood cells to do? Well, you should have got the fact that it causes them to produce antibodies. It can also cause them to produce antitoxins, but antibodies are the main driving bit that we're looking at. What happens if a pathogen infects your body after you've been vaccinated? Well, your memory white blood cells will be able to quickly produce the correct antibody and lead to the destruction of that pathogen so that you don't get ill. How do we describe someone who's been vaccinated against measles? We would say that they are immune to measles. And how can we reduce the spread of the disease? By vaccinating a large proportion of the population. So that is our herd immunity ideas. Right, make sure you've marked and corrected your answers, please. Right, in your booklets, there are tasks 2 and 3 on page 24 and 25 that we haven't done. I want you to do those, please. If you need some help, you've got there's the CGP revision guide, or there's Bite Size. There's a link on the Show My Homework for this, or you can use senecalearning.com to help you. Also on Show My Homework, there is an exam question that I would like you to have a look at. So download those and complete them for me, please. Right, in your booklets, at the top page 24, there was this activity, so just check that you have marked and corrected that answer, that sequence. Right, this is the evaluating use of vaccination, so task 2. So are they advantages or disadvantages? So first one, vaccinations help control lots of communicable diseases that are potentially life-threatening or have serious side effects. Of course, that's an advantage. The idea that vaccines don't work or that you need boosters, that's obviously a disadvantage. Preventing epidemics by vaccinating a large portion of the population is an advantage. Protecting those that cannot be vaccinated is an advantage. Painful, and you can have bad reactions, and you can, in very rare cases, you can have seizures and things like that. That's a disadvantage. And testing the vaccination on animals, and so the ethical things, is another disadvantage. Right, your diagrams of antibodies that would look a little bit like this one. Now, you might not have been sure what to do here, so I'll show you the first one and then you can pause the video and have a go at the second two. These were some quite difficult drawings to create on PowerPoint. So first one, what you'll be looking at, antibodies are these Y-shaped molecules. So we have these sites on the top which are going to be complementary, which these antigens are going to fit into. So you can pause the video now and have a go at the next two if you weren't sure about those ones. So next one would look like this. You might not have these side bits, you might just have a little sticky up bit there. That's fine. And then your third one, there should be a cutout, which of course, again, clearly we can see these things fit together with the shapes that we've got on the end of our antibodies here. So this is why we have different antibodies against a particular disease. And then the answers to the questions. So the reason that we're thinking about that different pathogens need different antibodies is, of course, the fact that different pathogens will have different shaped antigens. So antibodies will be specific to a specific antigen, so a specific disease and a specific pathogen. Therefore, we need different antibodies for different antigens and so different pathogens, because they are complementary and so they are specific. Therefore, a polio vaccination will produce polio antibodies, which will be against polio antigens, and the antigens in the rubella virus will be different. So therefore, the polio virus antibodies will not bind to the rubella virus antigens, so they will not protect against it. The reason that we won't be able to vaccinate against the cold virus is because the cold virus goes through, it mutates a lot. I think we mentioned this in lessons. And so when it mutates, we get different antigens on the surface. And so because it mutates a lot, it's very difficult to produce a vaccine that will work against it. Right, the exam questions. So I'll just take you through these. So the first one, you want to look at the fact that the vaccine is an active or a dead form of the pathogen. You could also use the term antigens instead. They stimulate antibody production. They can also stimulate antitoxin production. I wouldn't expect that one. By white blood cells, the antibodies will help to kill the pathogen. The antitoxins could neutralize the poisons. And so the antibodies will be quickly produced on reinfection because the memory cells are able to make them quickly. And you could also talk about that process of phagocytosis by white blood cells. And you need any three of those points in order to get 3 marks on that question. So the next one, was it a good investigation? No, because the sample size was small. There was only 12 people. The conclusion was based on hearsay from parents. It wasn't based on scientific data, and only 8 parents linked the autism to MMR. So it's a really small sample size, not very good. There was no control used. And so this is some of the ideas about why Wakefield's work was so poor. Was there bias? Well, yeah, because Wakefield was being paid by the parents or lawyers. So you can see that there were some real issues around the work that was done, and that's the work that a lot of anti-vaxxers use as a touchstone for reasons why vaccinations are poor. So question 2, we should be looking for antibodies out of the list, antitoxins and antibiotics, and then we need any 2 from measles, mumps or rubella. That's where the MMR term comes from. So measles, mumps and rubella. Now that was in the first one. You don't particularly need to do that, so don't worry too much. Right, that brings us to the end of today's lesson. Email me any questions, submit me some pictures of your exam questions please on Show My Homework. Hope you've enjoyed that. I will be looking at doing some live lessons with you next week, so keep an eye out for the announcements of that. Thanks a lot. Have a really good day, and I will catch up with you next lesson.